Hi, my name is Dr. Scarlett Raine, and today I'll be presenting our work, Automated Coral Spawn Monitoring for Reef Restoration, the Coral Spawn and Larvae Imaging Camera System, also known as CSLICS. Coral reefs are amongst the most biodiverse ecosystems on the planet, supporting 25% of all known marine life. However, they are at increasingly urgent risk, with some estimates projecting a 70 to 90% loss of coral reefs by the year 2050 due to increased ocean temperatures, coral bleaching events, and predation by the crown-of-thorns starfish. Reef restoration is the process of growing and replanting baby corals that are more resilient to warmer temperatures. We first collect spawn and bring it into the aquaculture facility, where we support that spawn to fertilize and become baby corals. We then grow those babies until they are big and strong, and then reseed them back onto the degraded reefs. This whole process is incredibly expensive, manual, and time-consuming. In this work, we tackle the coral fertilization stage, where we develop the world's first automated monitoring camera system to track the coral spawn and optimize tank conditions. Current spawn monitoring methods are extremely manual. Here we can see scientists extracting samples and then counting them under a microscope. The spawn is also incredibly fragile, so every time it is removed from the tank for counting, it can be damaged. CSLICS is a novel network of cameras and controllers mounted to aquaculture tanks. CSLICS has two operating modes: surface monitoring during fertilization, followed by subsurface operation over the subsequent 7 days. It performs real-time automated detection and counting of corals performed by YOLO V8 object detectors. The surface operation model detects the stage of embryo development over time, which allows us to assess fertilization success. We use an iterative process with a human expert in the loop for training these models. This graph shows the fertilization success rate increasing over time, and the bottom graphs show how CSLICS is able to detect an unsuccessful culture. These results are streamed to a centralized computer, allowing operators to remotely check all tanks in the CSLICS network. After the first 12 to 24 hours, CSLICS switches into the subsurface mode. The subsurface model counts the in-focus coral spawn over the following 7 days, enabling scientists to optimize tank conditions. This graph shows that the CSLICS counts align with the manual counts. After this period, the spawn are then moved to the next stage of the coral aquaculture process. CSLICS has been deployed to the Australian Institute of Marine Science, where it is in active use during coral spawning. We found that the system results in a 20-fold reduction in operation time as compared with manual counting, and this means a saving of over 5,000 hours of labour or $500,000 per spawning event, where we typically have 2 events per year. I'd like to acknowledge the work of the many co-authors and collaborators of this work, from the QUT Centre for Robotics, the Reef Restoration and Adaptation Program, and the Australian Institute of Marine Science. Thanks so much for your attention.